is Pratisha, and today we will discuss about photosynthesis, process of photosynthesis, and some of the experiment which prove that CO2, water, light, and green lamp is important for photosynthesis. So let's start. Photosynthesis. Photosynthesis is a word which is made up of two words. Photo, which means light. Synthesis, which means formation. So, what photosynthesis means is that in the presence of light, there is formation of a chemical. This is known as photosynthesis. By the word, we can say that. Now, in your NCERT, it is written that inorganic compound get converted into organic chemical or compound in the presence of light. This is known as photosynthesis. When inorganic compound like CO2, which is in gaseous phase, converted into C6H12O6 glucose in the presence of light, this process is known as photosynthesis. From this chemical reaction, we can see that from the CO2, C6H12O6 glucose is formed. What it means that CO2 get reduced because CO2 के पास तो हाइड्रोजन नहीं है, लेकिन ग्लूकोज में हाइड्रोजन है. So CO2 get reduced into the glucose. So the process photosynthesis is a reduction process. It is a reduction process where CO2 get reduced into the glucose molecule. Next, it is a physiochemical process. Physiochemical कैसे? As we can see that CO2 is in which form? Gaseous form, and glucose in aqueous form. जो ग्लूकोज बनता है प्लांट में, तो प्लांट की जो प्लांट सैप होता है, प्लांट सैप लिक्विड होता है जिसमें ग्लूकोज घुला हुआ रहता है और डिसॉल्व फॉर्म में रहता है. सो दिस ग्लूकोज in the aqueous form. Physiochemical process. Hence we can say that photosynthesis is a physiochemical process. Next one, it is an anabolic process. What is anabolic? It is a type of metabolism where any compound is synthesizing. When any compound is synthesizing in the presence of enzymes, that type of the process is known as anabolic process. So, in photosynthesis, there are various reactions we will study later, like light photosynthesis, dark photosynthesis, type of photosynthesis, where glucose, triose sugar, 3 carbon compound, 4 carbon compound are formed in the presence of various enzymes. Hence, this is known as what? Anabolic process, where enzyme is involved and any compound is synthesized. Next one, photosynthesis is an endothermic reaction. Photosynthesis is an endothermic reaction. What is endothermic reaction? Endothermic reaction are those reactions where any kind of energy is absorbed. Any kind of energy. It will be light, it will be heat. Where any kind of energy is absorbed in the reaction, that type of reaction is known as photosynthesis. So, if we will consider this process of photosynthesis, we can see that in the presence of light, and light is what? A form of energy. So, photosynthesis is what? Endothermic reaction, where energy is absorbed in the form of light. This is all about the introduction of photosynthesis, where we studied that photosynthesis is a reduction reaction, it is a physiochemical reaction, it is anabolic reaction, and it is an endothermic reaction. Next, we will study about the proof of photosynthesis. What is the proof of photosynthesis? Proof of photosynthesis. When any green plant photosynthesized, it will form first starch. It will form starch, glucose. Glucose will store in the form of starch. So that plant will show iodine test. Iodine test where the green part will show purple or blue color. This is the first proof that any green plant is photosynthesizing. The second proof is formation of oxygen bubbles. Formation of oxygen bubble. We will study it later that how oxygen is evolved from water molecule. But for this time we will see that कि what is the proof of photosynthesis? कोई भी plant, कोई भी green plant photosynthesize कर रहा है, तो it will form starch firstly, and if there is starch formation is occur in any plant, it will show iodine test. आयोडीन टेस्ट में जो ग्रीन पार्ट होगा जहां पे स्टार्च स्टोर हुआ है वो पर्पल और ब्लू कलर देता है. और any green plant if photosynthesizing, it will evolve what? Oxygen gas. So this is the proof of the photosynthesis. Next we will study that ये proof को बताने के लिए photosynthesis किसी प्लांट में हो रहा है, किस फैक्टर के थ्रू हो रहा है, इसके लिए बहुत सारे एक्सपेरिमेंट्स आए, बहुत सारे साइंटिस्ट ने इस पे वर्क किया कि CO2 is important, light is important, green plant is important, and more often सारे फैक्टर्स important है photosynthesis के लिए. तो बहुत सारे एक्सपेरिमेंट आपके एनसीईआरटी में मेंशन है. वी विल स्टडी अभी. नेक्स्ट, सो in class 10 we have studied that there is a variegated plant or leaf experiment. A leaf with two type of part. One part is green part, and other is non-green part. What it means? The green part has chlorophyll and it will form glucose which is stored in the starch, and non-green part does not have any type of pigment. So this type of leaf is known as variegated leaf, और ये commonly आप कहाँ देख सकते हो? मनी प्लांट में. मनी प्लांट में variegated leaf पाई जाती है. So the experiment is, सबसे पहले एक variegated leaf वाला प्लांट लिया गया. This is variegated leaf और उसको dark में रखा गया. In dark, what happened is that this green plant, this whole plant, the food, जो food बना था, जो starch बना था, because dark में कोई photosynthesis नहीं होती, तो सारा food क्या हो जाएगा? खत्म हो जाएगा. ठीक है? Food will finish. नेक्स्ट, इसी प्लांट को फिर हमने रखा कहाँ? लाइट में. इन लाइट. एंड एज वी नो इन लाइट फोटोसिंथेसिस विल अकर. तो फिर से फूड विल रीस्टोर दिस फोटोसिंथेसिस होगी तो स्टार्च बनेगा, फूड फिर से रीस्टोर हो जाता है। फिर इस लीफ को हम ट्रीट करते हैं विद द एल्कोहल। एल्कोहल से क्यों ट्रीट करते हैं? क्योंकि बेटा होता क्या है, देयर इज अ वेरीगेटेड लीफ, तो वेरीगेटेड लीफ में ग्रीन पार्ट विल ऑलवेज फोटोसिंथेसाइज एंड फॉर्म फूड। तो ये ग्रीन पार्ट से जो फूड होता है इट विल ट्रांसफर टू द नॉन ग्रीन पार्ट। दिस इज हैपनिंग इन अ वेरीगेटेड लीफ। हम एल्कोहल से ट्रीट जब करते हैं तो जो हमारा ग्रीन पार्ट होता है वो वहीं पे अपना फूड बनाता है और कोई ट्रांसफॉर्मेशन ऑफ द फूड ग्रीन टू नॉन ग्रीन पार्ट में नहीं होता है, ठीक है। एंड देन वी विल ट्रीट दिस alcoholic treated variegated leaf with iodine, and we will see that the green part will show purple color and non-green part will not show any type of color. By this experiment it was proven that starch is formed in the photosynthesis, glucose is formed in photosynthesis, which will restore in the form of starch in the plant by the iodine test. This is the experiment, ये आपने 10th में भी पढ़ा हुआ है। नेक्स्ट एक्सपेरिमेंट है वन हाफ का। नेक्स्ट इज वन हाफ एक्सपेरिमेंट। इसमें क्या था, देयर इज अ बीकर, बीकर में एक ऐसा लीफ रखते हैं which is some part of the leaf is in the beaker and some part of the leaf is outside the beaker, and then we will put a glass where KOH is put in the beaker। इस बीकर में KOH हम रख देते हैं। तो होता क्या है कि this green part will show iodine test, but this part which is in the beaker will not show iodine test। क्यों ऐसा होता है? In the photosynthesis or in the respiration, the leaf will respire and CO2 will remove, okay, and this CO2 is get absorbed by the KOH। This CO2 get absorbed by the KOH। Now अब CO2 है ही नहीं कि ये जो part of leaf which is present in the beaker वो photosynthesis कर सके, और जब photosynthesis नहीं होगी तो there is no production of starch, there is no production of glucose। Hence जो beaker है part of the leaf है will not show iodine test and there is no color of copper, but the outer part which is present outside the beaker will show iodine test because बाहर तो CO2 है, CO2 fix होगा, photosynthesis होगी और starch बनेगा। So by this experiment, इस experiment से ये conclude किया गया that is CO2 is important for photosynthesis। This is the experiment। ये दोनों experiment we have studied in the previous class 10। Now class 12 की experiment सबसे पहले है Priestley experiment। near about 35 near 1700 or 35। Priestley ने एक बेल जार लिया, there is a बेल जार, बेल type of बेल shaped jar जिसमें there is a candle which is burning and there is a mice। First, after few time, the candle get extinguished and the mice died because of the suffocation। Mice died because of the suffocation। In the next experiment, he had taken bell jar only, the candle, the burning candle, mice, and a green plant known as mint. He had taken a green plant, mint, mice, and burning candle. Now, he observed, after a few time, he observed that burning of candle is still going on, mice is not died, and plant is in the same position. By this experiment, the Bell-jar experiment of Whistling, he concluded that during the burning of candle, what is happening? Hydrocarbon, combustion of hydrocarbon is happening, and the CO2 is evolved from the hydrocarbon combustion. इससे क्या हो रहा है? और mice is also respiring. It will also evolve CO2. तो particular this bell jar, इस bell jar में CO2 का concentration बहुत high हो चुका है. The air in this bell jar get fouled. Because of the high CO2 concentration, mice get suffocated and died. In the next experiment, he had taken mint plant also. इसमें क्या हुआ? सेम वही चीज, हाइड्रोकार्बन इज बर्निंग, कंबशन हो रहा है तो CO2 is produced. Mice is also respiring to CO2 is produced, and all both of the CO2 from the candle and from the mice mixed in the bell jar. This both CO2 get restored or reabsorbed by the plant, mint plant, for their photosynthesis process. So photosynthesis के लिए ये CO2 absorb कर ले रहा है mint plant और वो O2 रिलीज करते रह रहा है, जिसकी वजह से माइस की भी डेथ नहीं हो रही है और ऑक्सीजन इज रिक्वायर्ड फॉर द कंबशन. सो कैंडल भी बर्निंग ऑफ द कैंडल भी स्टिल गोइंग ऑन है, माइस की सफोकेशन से डेथ नहीं हो रही है बिकॉज ऑक्सीजन का कंसंट्रेशन है बिकॉज ऑफ द मिंट प्लांट. फोटोसिंथेसिस में वो सारे CO2 को एब्जॉर्ब करके O2 निकाल रहा है. ठीक है, तो बाय दिस एक्सपेरिमेंट he proven that a green plant is important for photosynthesis. If there is no green plant, there will not photosynthesis happening. This is Priestley experiment. Next experiment is of Eganhouse. House experiment. Yes. Eganhouse experiment. He had taken an aquatic plant. Aquatic plant. This is an aquatic plant. Its name is Hydrilla. और फिर इन्होंने सोर्स ऑफ लाइट, आर्टिफिशियल सोर्स ऑफ लाइट से, दिस इज सोर्स ऑफ लाइट, इस प्लांट में लाइट आई. सो द रीजन वेयर हाई लाइट intensity is falling on this hydrilla plant, this aquatic plant. It is observed that bubble of oxygen is formed at that area. Eganhouse had taken an aquatic plant, hydrilla, और फिर उन्होंने उसमें एक आर्टिफिशियल सोर्स ऑफ लाइट से लाइट गिराई, and he observed that the region where light is in high intensity, oxygen bubbles are formed. By this experiment, he proven that oxygen is evolved or released during the process of photosynthesis. This is the experiment of John Eganhouse. Next experiment we will study is von Sack experiment. Von Sack's experiment. One-set experiment, he said that CO2 get reduced into the C6H12O6. He said that photosynthesis is a reduction process where CO2 get reduced into the glucose. And he also observed that there are some green areas of pigment which is later said that it is what? Chlorophyll. And some green structure present in cell which is later known as chloroplast. He also observed that the green area and the green structure which is present in the cell will fix hydrogen, or we can say that CO2 get reduced into the C6H12O6 at this green area and the green pigment. Later it will study that the green area is the chlorophyll and the green cell or green structure present in the cell is chloroplast. This is all about the one-sack experiment of the photosynthesis. Next experiment we will study is Engelmann TW Engelmann experiment. TW Engelmann experiment. He took a green filamentous algae known as cladophora. We had studied about the algae in our previous class that green algae are those which have chlorophyll A, B, and some of their accessory pigment like carotenoid, and the result food material of this green algae is starch. Some green algae are unicellular and filamentous. So filamentous algae is cladophora, and the suspension have aerobic bacteria also. Aerobic bacteria. He had taken an suspension of filamentous algae, green filamentous algae, filamentous green algae, cladophora, and some of the aerobic bacteria. Okay. Then there is source of light. Source of light. जो source of light prism में आती है, and as we have studied in the class 10 कि जब भी कोई source of monochromatic light होती है और prism पे आती है तो the prism will scatter the light. Okay, तो prism क्या करेगा, उस light को scatter कर देता है. Okay, in the form of Vibgyor. This is the scattered light. So he observed that जो red light वाला region है और blue light वाला region है जो इस cladophora पे fall हो रहा है, blue और red light वाला region, वहाँ पे aerobic bacteria get accumulated in large amount. As we can see that in this is the red light region जो fall हो रही है on the cladophora green algae, वहाँ पे aerobic bacteria ज़्यादा accumulate होते हैं, and in the blue light region वहाँ पे भी aerobic bacteria ज़्यादा accumulate होते हैं. By this experiment, अब इस experiment से हम क्या देख सकते हैं that red light and blue light region जो होता है वहाँ पे intensity of photosynthesis या amount of photosynthesis is higher as comparison to the other light, like orange, yellow, green and violet. This light region से blue and red light region में तो जो photosynthesis की जो rate है वो थोड़ी ज़्यादा है. इससे ये पता चलता है क्योंकि यहाँ पे जब फोटोसिंथेसिस ज्यादा होगा तो ऑक्सीजन का इवोल्यूशन रिलीज ऑफ ऑक्सीजन भी ज्यादा होगा, और जब ऑक्सीजन ज्यादा होगा तो वहीं पे एरोबिक बैक्टीरिया है, एरोबिक बैक्टीरिया मतलब ऐसे बैक्टीरिया जो ऑक्सीजन की प्रेजेंस में रेस्पिरेशन करते हैं या ऑक्सीजन की प्रेजेंस में अपने ब्रेकडाउन मतलब रेस्पिरेटरी सबस्टेंस करते हैं, तो वहां पे जाके एक्यूमुलेट हो जा रहा है, जिससे ये पता चल रहा है कि रेड लाइट और ब्लू लाइट रीजन में इंटेंसिटी ऑफ द फोटोसिंथेसिस या अमाउंट ऑफ द फोटोसिंथेसिस ज्यादा है। ओके, दिस इज द एक्सपेरिमेंट ऑफ टी.डब्ल्यू. एंगलमैन, एंड टी.डब्ल्यू. एंगलमैन ने सबसे पहले, he was the first one who said Action spectrum. Action spectrum. He said the name, or the we can say the word of action spectrum. Now we will study about action spectrum also. There is a graph. Okay, we will plot a graph. यहाँ पे होगा light, range of light, and this is action spectrum. The word action spectrum, इसका मतलब क्या है? Action is function, और simplify किया जाए, this is work. And what is work of green plant? Photosynthesis. So it is photosynthesis. और spectrum is what? Range of light, जो विजिबल रेंज में आपकी लाइट्स होती हैं, विजिबल वाली वो, ठीक है। तो एक हमने प्लॉट बनाया है जिसमें हमने एक्स एक्सिस पे क्या रखा हुआ है, एक्शन स्पेक्ट्रम, और वाई एक्सिस पे क्या रखा हुआ है, रेंज ऑफ लाइट। You observe that the action spectrum get peaked at the range of red and at the range of blue. This is action spectrum. रेड और ब्लू रीजन में जो फोटोसिंथेसिस की जो रेट थी प्लांट की, वो इंक्रीज हो जाती है, ठीक है। और उसको जब उन्होंने प्लॉट किया, एक प्लॉट बनाया, एक ग्राफ प्लॉट किया, जिसमें उन्होंने रेंज ऑफ लाइट रखी एक साइड पे एक्शन स्पेक्ट्रम रखा, देखा कि ब्लू और रेड लाइट रीजन में फोटोसिंथेसिस की रेट क्या हो रही है, बढ़ रही है, व्हिच इज नोन एज एक्शन। सो दिस इज द एक्शन स्पेक्ट्रम। नेक्स्ट एक्सपेरिमेंट was given by Cornelius Neal experiment. So earlier it was said that CO2 get reduced into the C6H12O6. ये बोला जा रहा था कि this CO2, this is water, और इससे C6H12O6 बन जाता है। This is reduction process. Cornelius Neal ने बताया कि if in the place of H2O there would be some more compound like H2S in some green sulfur bacteria and purple bacteria. These are the bacteria, ऐसे kind of bacteria हैं जो कीमोसिंथेसिस करते हैं। कीमोसिंथेसिस का मतलब केमिकल से अपना फूड बनाते हैं, ठीक है। तो green sulfur bacteria, purple bacteria are those bacteria who synthesize their food from the chemical. उन्होंने देखा कि in the place of H2O, अगर हम H2S रखते हैं तो S in the free form रिलीज होता है, in the place of O2, जहाँ पे O2 रिलीज होता था यहाँ पे S रिलीज होगा, और this C6H12O6, or we can say that CH2O, this is formula for carbohydrate will form. अगर ऑक्सीजन की प्लेस पे आप H2S रखते हो, और ऐसे H2S रखने वाले या फिर ऐसे H2O के अलावा कोई और केमिकल से CO2 को रिड्यूस करने वाले बहुत सारे बैक्टीरिया होते हैं, इसमें से ग्रीन सल्फर और पर्पल सल्फर बैक्टीरिया का एग्जांपल दिया हुआ है आप लोगों ने। तो दिस इज द एक्सपेरिमेंट। नेक्स्ट वन, radioisotopes. What is radioisotopes? Radioisotopes. O816 ये होता है ऑक्सीजन का एटॉमिक नंबर एंड एटॉमिक मास। This is radioisotope of oxygen. Okay. So he had taken radioisotope of oxygen in H2O. In H2O में ऑक्सीजन जो है वो 18 वाला है, इट मींस इट इज अ रेडियोएक्टिव आइसोटोप एंड दिस CO2 in the presence of light C6H12O6 will form glucose plus 6O2 plus 6H2. It was proven by this experiment, it was proven that the oxygen which is evolved during the photosynthesis, released during the photosynthesis, will come from the H2O, not from the CO2. Okay? फिर से हम देख लेते हैं। This is the experiment जिसमें उन्होंने रेडियो आइसोटोप ऑफ ऑक्सीजन लिया, H2O का जो ऑक्सीजन था वो रेडियो आइसोटोप लिया। What is रेडियो आइसोटोप? This is isotope and this is radioisotope. Okay. फिर he had taken H2O, 6 CO2 in the presence of light, उन्होंने देखा कि ग्लूकोज बनता है C6H12O6 एंड ऑक्सीजन मॉलिक्यूल रिलीज होता है जिसमें कि सिर्फ जिसमें O18 आया। इट मींस ऑक्सीजन जो इवॉल्व होता है या जो रिलीज होता है ड्यूरिंग द फोटोसिंथेसिस will come from where? H2O, not from the CO2. And the CO2, for CO2 get reduced into the C6H12O6 glucose. So this is all about the experiment जो कि प्रूव करता है कि CO2, water and green plant, light is important for photosynthesis. Some of the scientists have proven that by this experiment, all the factors are important for photosynthesis. This is all about the class. Thank you.